I really want to thank Peter for the opportunity to give an updated version of a talk that I gave, at least part of this talk I gave in Poland a couple of years ago, nearly 18 months maybe, at the European Leadership Forum, where we looked at what makes us human, genetic evidence, and I spoke about various things and then focused on this issue, which is ancient man. I just want to reiterate the point that I'm not a geneticist, but I'm obviously, like everybody, interested in this particular subject. I am always encouraged when I have to give some sort of technical talk, that all of a sudden, in the days leading up to the talk, there's something published, even through it was through the BBC website that I actually came across this work. That was on the BBC website of the 7th of January. That was the day before I left Israel. And just one quote within that was on the BBC website. Our big surprise was to find that this gene region has such a high Neanderthal ancestry because this region has been shown to have major biological relevance in host survival against pathogens. It's quoting this work, actually two papers, American Journal of Human Genetics, that went online on the 7th of January 2016. And a second paper actually in the same journal which followed on, which is looking at immunity and genes associated with immunity, where these Neanderthal signatures were very strongly seen. I have to put this one in as well, because I found this one, I think it was this morning, or was it yesterday, I can't remember, which I did send out to the few people whose email addresses I had available. Ancient DNA and the Rewriting of Human History. Be sparing with Occam's razor. I'll mention that again a bit later, but of course it's very interesting to realise that this particular paper in Genome Biology comes out of Cambridge. It's just up the road, and I assume that Matt will know most of these authors, I would assume. Again, I feel a cheat or a fraud really, because in a sense Matthew should be giving talks like this, I think. It mentions ancient DNA in the title, which of course is good, and I could have called my talk Ancient DNA, I suppose, but I wanted to make it a little bit more complete by looking at the discovery of Neanderthal man very briefly, and the distribution of Neanderthal man. We might need to redefine that a little bit. But I then do want to look at what the academic world is saying about the DNA of Neanderthal man. It's a talk in 3D. Okay? Shows that I'm a Baptist preacher, really. 3-point sermon, alliteration and all that sort of stuff. But actually, I want to go on to say something at the end of the talk. What does the Bible say about ancient man? So if we look at the discovery of Neanderthal man, we need to go to the river. This is a river near Dusseldorf. It's in a valley. The valley is called the Neanderthal or the Neander Valley. Interesting to note that it's actually in a cave, the Fieldhofer Grotto, where Neanderthal man was first discovered in 1856. It's an interesting date. Not a complete skeleton, a skull cap where you see the brow ridge, you see the very substantial, very robust skeletal remains. Discovered in 1856, which of course is interesting because it's 3 years before the publication on the origin of species, and it is worth noting that in that book Charles Darwin avoided the subject of human evolution, although he did say this: light will be thrown on the origin of man and his history. He was to go on and write another book, The Descent of Man, which was published in 1871. I suppose the interest in Neanderthal Man exploded around the year 1908, where a nearly complete skeleton of Neanderthal Man was discovered in France, with its very large brain box, which presumably means a large brain inside. And also, again, the very robust frame when you compare the skeletal remains with modern humans. And of course, since then, Neanderthals have been discovered in very many different places. There's a distribution, and here I list 4 different places where Neanderthal remains have been found. We'll mention 1 or 2 of these a little bit later in the talk. Well, one of the things that we need to note, of course, is that they're all in caves. I suppose we're thinking of the preservation of ancient DNA. It needs to be kept cold as well, found in caves. Well, ask the question, why this distribution? And I think it's quite interesting to use this map, because I think one can overlay over this map the extent of the ice during the Ice Age, which of course limits the areas where the Neanderthal would have settled. Okay, I'm going to say a bit more about distribution a bit later, but I want to really talk about something that is so current and so interesting, the DNA of Neanderthal Man. And we need to go back to May 2010. This was shortly before one of our European Leadership Forum summits in Poland, where the group in Germany published a draft sequence talking about 4 billion nucleotides, but actually from 3 individuals. It wasn't a complete, it was only draft. But what they did show in this work was that the Neanderthal shares genetic variants with present-day humans in Eurasia, rather than present-day humans in sub-Saharan Africa. And this is where the issue of Africa comes and hits us in the face again. So really, just to summarise that in a way visually that we can see, Neanderthal signatures are found in Eurasians. Well, that's what was said at that particular time. And here, again on the BBC website in May 2010, we saw something like this. Family tree, if you like, or ancestry, expressed diagrammatically. Interestingly, it talks about Neanderthals dying out here, but with some Neanderthal and modern humans, Homo sapiens, interbreeding. Introduces this word, which, to be honest with you, I don't like the word, I have to be honest with you, admixture. What does admixture mean? I think it is an obfuscation. Is that an English word? Mm-hmm. It's trying to hide something that I think becomes absolutely clear in due course. But now it gets more complicated. The same year, 2010, we had this publication in Nature, The Genetic History of an Archaic Hominin Group from the Denisova Cave in Siberia. And here's part of the abstract. A finger bone found in the Denisova Cave in southern Siberia, I think it's in the Altai Mountains. Here you see again that it's a cave. And here you see a little map where we located in Siberia. And here is another picture of some of the work to uncover some of the skeletal remains. This is the finger bone, or at least a replica of the finger bone. So it's not a big piece of bone. And what does it say? Let's highlight a few words. This individual is from a group that shares a common origin with the Neanderthals, but not involved, this is what they're saying in 2010, not involved in the putative gene flow from Neanderthals into Eurasians. Alright, we've just seen that this is a suggestion that was made in the earlier paper, However, the data suggests that it contributed 4 to 6% of its genetic material to the genomes of present-day Melanesians, and so that's just the summary visually presented to us. But again, we see that now it gets much more complicated, because we're now moving to 2014, and here we're getting a much more complete genome sequence of a Neanderthal, but from the Altai Mountains. In fact, in the very same place. This time, a proximal toe phalanx was recovered in the east gallery of the Denisova cave, number 3. Layer 11, where both the finger and, that's what we've talked about thus far, the finger, which we got the Denisovan signature, and the toe phalanx were found, thought to be at least 50,000 years old. The finger was found in a sublayer, 11.2, whereas the toe derives from a lower sublayer, 11.4, and may thus be older than the finger. You know, for me, it seems to me that this is clutching at straws. From the same paper, of course, what do we say? An analysis of the relationships and population history of available archaic genomes and the 25 present-day human genomes show that several gene flow events occurred among Neanderthals, Denisovans, and early modern humans, possibly including a gene flow into Denisovans from an unknown archaic group. But now it gets even more complicated. This is actually just a few months, I think, later. We determine an almost complete mitochondrial genome sequence of a hominin from Cima de los Huesos, and show that it's closely related to the lineage leading to mitochondrial genomes of Denisovans and Eastern Eurasian sister group to the Neanderthals. Now, this is again another picture of another cave, but this is Cima de los Huesos, which actually in English means the pit of bones. And here we see a map where we see it located. Closely related to the Denisovans. This is interesting. Okay, so here we've got the Denisova cave, and here we've got the cave which is Cima de los Huesos. But this is mitochondrial genome, so really I want to say a little bit about the mitochondria because I find it incredibly interesting. Mitochondria are these little organelles in the cytoplasm that do carry their own DNA. Not very big. It's a circular molecule. The fact is there's about 100 in some cells, but up to 10,000 mitochondria in other types of cell. This, to my mind, is incredibly interesting. The human mitochondrial DNA codes for only 13 proteins. But actually, the mitochondria needs over 1,000 proteins. Where do the others come from? They're all coded for in the nucleus. The rest of mitochondrial DNA codes for essential RNA molecules. Some transfer RNA molecules, some ribosomal RNA molecules. This, I think, is incredibly significant because this DNA is in the mitochondria. It doesn't benefit from all of the incredible repair machinery that is available in the nucleus. Therefore, it is more easily mutated. This, I think, is very significant. You need to remember this, because we're going to come back to it, okay? So, okay, what can we say? There's some connection between these two. And because we've already come to this conclusion, Denisovan signatures in Melanesians, we can extend, because we've got the support of this paper, investigating the evolutionary importance of Denisovan introgressions in Papua New Guineans and Australians. That was published online in July 2015, so we're talking relatively current. But there's another paper, actually it was published a little bit earlier, Denisovan ancestry in East Eurasian and Native American populations. So we can extend these connections even further. And actually this is exactly what the authors say in their conclusion. In any event, the inescapable conclusion is that Denisovan ancestry is more widespread in modern human populations than thought previously. This is going back, only a few years, to be the case. And moreover, human genetic history must consequently also be more complicated than previously believed. Of course, we've still got this problem to solve, the Africa problem. And here's another paper. The genomic landscape of Neanderthal ancestry in present-day humans. Genomic studies have shown that Neanderthals interbred with modern humans and that non-Africans today are the products of this mixture. Okay, so you see the isolation of Africa in all of this. Anyway, let's think about this for a moment. The evidence of ancient DNA from 2 or perhaps 3 sources is found in the modern human genome. Exactly what does this mean? Are we talking about different genes on different chromosomes? Well, it actually depends on the definition of the gene. We're talking, and we've already used this word, of course, today. We're talking about different alleles, which are actually involved in the variation of gene expression. There's so much confusion, it seems to me, out there as to what a gene and an allele is. However, going back to this paper, it does say this quite accurately. The paper published in 2014, regions that harbour high frequency of Neanderthal alleles are enriched for genes affecting keratin filaments, suggesting that Neanderthal alleles may have helped modern humans to adapt to a non-African environment. So you can see here the background assumption is that the humanity has come out of Africa. Goes on to say this: we identify multiple Neanderthal-derived alleles that confer risk for disease, suggesting that Neanderthal alleles continue to shape human biology. This is 2014. In other words, different alleles. What's an allele? Actually must include the non-protein coding regions. Certainly that which binds promoters and transcription factors will affect the expression of any given gene. And I felt it helpful in my previous talk and in this one to give an example, because I think it highlights where there is confusion over these issues. If we think of brown eyes and blue eyes, we're talking about the expression of melanin in the iris, and here is a little pathway which leads from an amino acid, tyrosine, with an enzyme, tyrosinase, through dopaquinone, which is then non-enzymatically converted to melanin. Here you see the structure of melanin. It's not a protein. Melanin. So it doesn't have a protein coding gene that leads to melanin. There is a protein coding gene that leads to tyrosinase. If there is a low expression of melanin in the iris, we get a blue eye. If there is a high expression of melanin, we get a brown eye. What expression are we talking about? We're talking of the expression of this particular enzyme that converts tyrosine to dopaquinone, which is an intermediate before melanin. However, what does mutation do with respect to this? It actually interferes or even prevents the action of tyrosinase. So what do we get? We get albinism. The expression of melanin in the iris has got everything to do with epigenetic modification, and I thought I'd put this in too because it leads to something else with respect to the Neanderthal. Talked about this also at the European Leadership Forum. Methylation generally closes the chromatin and prevents transcription. Many different factors, but including demethylation and acetylation, opens up the chromatin and then you get transcription. The actual methylation process, or the one that is now of interest to us in this particular talk, is where you get the conversion of one of the bases, cytosine, to 5-methyl cytosine. Why am I bringing that into my talk? Well, it's basically because of this work, which was published also in 2014, reconstructing the DNA methylation maps of the Neanderthal and the Denisovan. Now, I put somebody's face in here because this is Liran Carmel, who's of the Department of Genetics, the Hebrew University in Jerusalem, and I mention him because when I went to this genome conference on behalf of Paul Nelson initially, Liran Carmel actually presented this work. I won't go into all the details, but of course archaic DNA over time naturally deaminates, and there's a differential deamination. If we're talking cytosine, deamination leads to uracil. Alright, the deamination is the removal of that bit. If we're talking 5-methyl cytosine, which would be present where there has been methylation of the DNA, altering its reactivity in particular areas, the deamination, the natural deamination, leads to thymine. Now you can remove uracil chemically, and thus you can go back to the archaic DNA to demonstrate where it has been methylated. It's technically complicated. We identified approximately 2,000 differentially methylated regions, particularly in the HOXD cluster, which may explain anatomical differences between archaic and present-day humans. And here you see just a comparison of the Neanderthal skeleton with a modern human, and you can see a larger brain box and a much more robust frame. So what do we think we know? Well, possibly, on the basis of what's published in the academic literature, there are 3 distinct groups of ancient man: Neanderthals, Denisovans, and a third unknown group. And modern man carries different signatures of these groups. So here's my question: exactly how much? And this brings me to this paper, and if there's a godfather with respect to Neanderthal and Denisovan DNA, it is this man, Svante Pääbo. Am I pronouncing it correctly, Ola? Pääbo. Yeah. Do you know him?
No, I've heard him lecturing once.
Right.
But he's,
The contribution of ancient hominin genomes from Siberia to our understanding of human evolution. This was published actually in a very obscure journal, but in 2015. It's worth reading what he writes here. The high quality of the Neanderthal genome from the Denisova cave can also be used to estimate what parts of the genomes of present-day people were inherited from Neanderthals. This confirms that everybody, we're talking individuals here, this confirms that everybody outside sub-Saharan Africa, here we go again, carries between 1 to 2% of Neanderthal DNA. Now, all of us are carrying at least 1 or 2% of DNA which has come to us eventually from the Neanderthal. And actually, if you get your DNA sequenced, and I do want to get it done, it doesn't cost very much. I mean, it's amazing when you think about it. The Human Genome Project took almost billions of pounds, tens of years, thousands of man years to get the draft of the human genome. You can get your genome sequenced now for something like $100. And you get from 23andMe, which is a company that does such things, amongst a lot of other things, a Neanderthal score, which is quite interesting. This is what Svante says. To get a perspective on this, now think about this, folks. This is what he says. I'm just lifting it out of his paper. You may recall that we all have one half of our DNA from each of our parents, about 25% from each grandparent, about 12% from our great-grandparents, and so on. From an ancestor 6 generations back, we have on average inherited about 1.5% of our DNA. Thus, not my words, from the point of view of the total amount of DNA people today have inherited from Neanderthals, it is as if they had a Neanderthal ancestor 6 generations back. Now that's an interesting statement for him to make. I've got the question mark. What are you talking about here? Well, of course he uses then the word however. But this is what he says. For me it's fantastic. Due to recombination that occurs when new germ cells are formed in each generation, the Neanderthal DNA is distributed in much smaller fragments than the DNA you have inherited from your ancestors 6 generations back. Okay. However, highlighting words, recombination and new germ cells, because this is one of my... I love talking about meiosis, and we need to look just briefly at meiosis. In other words, the formation of the germ cells, sperm and egg. Of course, when we're talking humans, parent cells have 23 pairs of chromosomes. Here you see 46 individual chromosomes. But in the process of meiosis, you get a reduction division, and sperm and egg have half that number, so we have 23 individual chromosomes. It's going to be an X in the ovum, but of course you'll have an X or a Y sperm. Of course, at fertilisation you get the reversal of fortune, and we get back our 23 pairs of chromosomes. Meiosis is an incredible phenomenon. It doesn't begin until you get the pairing of homologous chromosomes. Yes, they'll have different alleles, that's for sure, but structurally they're essentially identical. And the pairing of chromosomes, well, what can we say? Well, we get 2 copies of each chromosome. One originally derived from our fathers, one derived from our mothers. Meiosis doesn't and cannot start until the pairs are zipped together. X and Y is interesting because there is a small region of where the Y chromosome has comparable regions on it to the X. Most of it's very different, but there are at the very extremes things that are a little bit X-like. That's why people want to say that the Y chromosome has actually come to us by a devolved process from something that was originally an X. We don't need to go into that one today. The fact is, synapsis is extremely complex and accurate, and here's a fantastic picture of something that is called the synaptonemal complex. Our individual chromosomes from our father and from our mother are already in the process of dividing. However, they're zipped together in a very specific way. I just wanted to change this. Everybody accepts that there's admixture. That means there's interbreeding. And what I can conceive in my mind is that you may have a Neanderthal father, I'm going back in history of course, and a modern, and I'm putting those in inverted commas of course, a Neanderthal father and a modern mother. Here's another picture of the synaptonemal complex, which is from Nature Reviews in Genetics. This is some time ago, in March 2009, where it says it has been known for decades that errors in chromosome synapsis, that is asynapsis, are associated with impaired fertility and with high levels of asynapsis sterility, which is seen in both sexes. The second stage of meiosis is the recombination process, and this is a fantastic cartoon, or whatever you want to call it, animation, where you see the crossing over. This is, I think, technically called a holiday junction. And it always seems to me to be remarkable because you're getting crossing over, but something has to be extremely precise here. Is mutation at all tolerated where there's recombination? I'm not sure if there can be. Of course, we're talking haplotypes and recombination hotspots. It has to, if you're getting this crossing over prospect, it requires total and complete alignment of homologous chromosomes. It's not ad hoc. This is so carefully orchestrated. It involves a careful check on the integrity of the DNA sequences at recombination hotspots. This couldn't happen ad hoc. I doubt whether mutation can actually be tolerated here at all. It's the question, isn't it? You know. Something to think about. Meiosis cannot begin without synapsis. I'm just looking at the time. And recombination. Synapsis cannot happen without the maintenance of chromosomal integrity. If there's no meiosis, there's no sperm or egg. If there's no meiosis, there's no survival. But we're here, folks. All of us have got Neanderthal scores. What does it mean? Now, let's go back to this paper. Let's just read what he says. You may also ask how much of the total Neanderthal genome exists distributed among people living today. We talked of individual content, 1 to 2%. This estimate is still very approximate, but it would seem that at least about 40% of the Neanderthal genome can be found in people today, if we're thinking collectively. This is an amazing statement to make. Let's think about it. Distributed, at least about 40% of the Neanderthal genome is found distributed in people today. What else can we say? Neanderthals and Denisovans coexisted, cohabited, and interbred with, in inverted commas, modern man. But this is the point. They produce fertile offspring. There's no issue of sterility here at all. Why? It's obvious. They are fully human, albeit ancient.